hopefully you've had a chance to watch the importance of proteins video to give you an idea of just how important proteins are. In fact, when we start our genetics unit, you're going to see just how important proteins are because that entire unit revolves around figuring out how our bodies make proteins because they make up everything in our body. They're part of all of the cells in our body. They're part of our hair, our nails. Everything in our body contains protein, so it has to be the most important and diverse. So there has to be lots of variety of proteins in order to have all of those different functions. And the way that they do that is because they're large groups, remember the word poly meaning many, of smaller subunits called amino acids. So this is important. Proteins are made up of smaller subunits called amino acids. And they're joined through the condensation reaction. Remember, we just dealt with the formation of our triglycerides. Now, proteins, because we have so many and they have so many important jobs in our body, their shape is really, really important, and it determines their function. So if their shape changes at all, then the function of that protein may no longer work and may no longer do its job. And there's lots of things that can actually disable proteins from doing their job, which we'll look at some examples of. And their shape in general is actually determined by the types of amino acids. So I think we should start with the basics first. Let's go to the very basic subunit of a protein, is an amino acid. So there are 20 different types of amino acids. And just to give you an idea, since I don't have them all here, but your textbook does list them all, in your textbook on page 42, and this highlighter is not going to do this for us. So, okay, got my pen back here. Just so you have reference to all the different amino acids to have a look at the variety that we have of smaller subunits that we have to start with, we're on page 42 in our textbook. So there's 20 different amino acids total, but here's what's similar about every single one, and here's why we're outlining the basic amino acid structure. Every single amino acid, all 20 of them, will contain this backbone that you see here, and I'm going to highlight the importance of what you see here. So again, remember this R just means that other groups of molecules and atoms can come off of this end of the carbon. Remember, carbon has 4 bonding sites. These 2 are taken up, this one's taken up, and so here's where we add different groups to make each amino acid unique to itself. And this is how we get our 20 different ones, is by changing what comes off of this carbon right here. So the basic structure of every single amino acid will contain these things. So number 1, this group here, hopefully you remember what this group is the amino group. And on the other side of that carbon, the other end of your amino acids, because they're at opposite ends, we have this group. Remember the functional groups we talked about. This is your carboxyl group. So every single amino acid will have these 2 groups in common with carbon in between them, and it's this group here that's going to change that make up our 20 total that we have. Okay? And one other important thing, I'm going to highlight this. This is very important. Out of the 20, 8 are considered essential. So what that means to us is that we can get them from eating different foods. The other remaining 12, we can't eat them, but they are constructed by our body. And so the word essential is what means we can obtain from diet or the foods that we eat. Okay, next, so if we know that proteins are made up of a whole bunch of smaller subunits called amino acids, then the next thing we need to understand or know is how do all of these amino acids join together to make up these proteins. So that's our next step. Formation of a peptide bond. A peptide bond is what's formed when amino acids come together. So right now, I just want to highlight when we have any 2 amino acids, and again, it doesn't matter what amino acids there are. This group will differ depending on the amino acid, because the only thing that matters when a peptide bond is forming is the alignment of these 2 groups. Okay, and I'm going to highlight these again. So remember, our carboxyl group at one end of an amino acid comes near our amino group of another amino acid. And this is where our peptide bond is going to form. So if we look, you might actually already be able to predict based on what's highlighted here for you. It has to be one of the 4 reaction types we learned about. So if you recall, in a condensation reaction, one molecule has to give up an oxygen and hydrogen, and the other molecule has to give up a hydrogen. So you can see here, this guy is going to give up his oxygen and hydrogen, and this guy is going to give up his hydrogen. So these two end up in water. And what's left when that water comes out always is the remaining molecules will come together and join. So you can see what's left is a carbon double bonded to an oxygen, and that's right here. And then this group has a nitrogen and a hydrogen, which is right here. And the peptide bond is what forms in between those two. So I want to highlight, I want to highlight the peptide bond itself. The actual bond, they've highlighted in green. I'm going to make it even darker. So this is our peptide bond. It is the bond between the carbon and nitrogen in this group, and I'm going to highlight this. Once they've come together, right here, let's use black. Here are the atoms involved in our peptide bond. So a nitrogen and a hydrogen, a carbon double bonded to an oxygen. These are the atoms involved in our peptide bond, always between 2 amino acids, and this is what we call the actual peptide bond, the one between the carbon and the nitrogen. Okay, now that we know how amino acids come together to make up proteins, we're going to look at the different categories of proteins based on how many amino acids and the shapes of the different molecules that they take on. They can get put into a few different categories. So that first category, the most basic one, we call the protein's primary structure. This is just a chain of amino acids linked together in basically a linear chain. So just one straight line of amino acids linked together in those peptide bonds we just showed. And they call that a polypeptide chain, because it is just a linear chain. It's not branching, it's just one straight thing. And what's determined, it's determined from codes in our genetics. That's why I related at the beginning of this note that when we talk about how important proteins are, there's a whole unit devoted to the production of proteins in our body, and that has to do with the codes that are in our genes. Okay, and our secondary structure is the next category of proteins. So we're starting out small with our primary structure, and we're getting more and more complex as we go. So secondary structure can be one of two things. Alpha helix, and this is, remember when we talked about the types of bonds, hydrogen bond being one of them? If a hydrogen bond exists between the carboxyl and amino group of linear polypeptide chains, so if we took just the primary structure, one chain of amino acids, and it were to then, if you look here, here's our carboxyl group. I'm going to use a pen here. Here's our carboxyl group. So once that peptide bond's formed, here's our carbon. So this is a carbon, and this is an oxygen. And on that same chain, we've got our nitrogen and our hydrogen here. And if you recall, the definition of a hydrogen bond is a hydrogen of one molecule interacting with the electronegative end of another molecule. So this actually takes your polypeptide chain that you formed in your primary structure, and it starts to bend it a little bit, because this hydrogen bond forms between the hydrogen on the amino group and the oxygen on the carbon group. So you end up getting what they call is a helix, sort of this ribbon structure here. And that's the name they give it. Alpha helix is one possible structure that falls into the category of secondary structures. And then the second one is called a beta-pleated sheet. Now, if you go back and recall, the primary structure are these just single chains of polypeptides. And all that a beta-pleated sheet is when these two single linear chains lie near one another, they interact and they end up forming this shape here. And it looks like an accordion, and they call it a beta-pleated sheet. So those are the two possible secondary structures of proteins. And as you can see, they get a little bit more and more complex in terms of their shape as we add more um, rows of polypeptide chains, or they start to bend. This is all that's really happening. They are still made up of amino acids. They're still made up of the 20 that we have to choose from. Okay, the last two groups are really just short explanations, but the picture says more actually. A tertiary structure, if the polypeptide chains just continue bending and folding on itself, they just make up a very three-dimensional complex structure is what we call the tertiary structure. In the quaternary, so the final subgroup of types of proteins, is basically now, if we look back at our tertiary, this was more complex because they continued to fold and bend and new forces were existing between the polypeptide chains. So if more than one of these were to come together, we can actually make an even more complex protein, and they call that quaternary. So it's the clustering of several polypeptide chains that eventually take on a specific shape. Now remember, the whole basis behind proteins and what they do is determined by its shape.